Okay, so today is just for the record again, I guess in case you fast forwarded, we're going to start recap of lesson 1-2, which is the examples. Now, this stuff gets a little bit technical. I do suggest that you either watch my video or you watch the one from Amoeba Sisters or you watch the Bozeman Science one, but do watch something about this because it's technical. So I'm going to take some time, I'm not going through all of these. You can read that all about that in my very, very exciting video on this that I canned for you and put in the content. But I need you to understand how the lac operon is arranged on the DNA strand. First of all, the lac operon is a collection of genes that codes for what? Who watched my video, stroke my ego, and tell me, what is this collection of genes coding for?
Breaks down lactose.
Good. What does?
The
Lactate.
enzyme.
Yep, the gene enzyme. Okay, great. Right. Yes. So this little section here is 3 different genes. Do we see that? Let's zoom in. Woo. This is 3 different singular sections of the DNA strand. Okay, this is DNA. It doesn't have the double helix. Sometimes artist's interpretation looks a little different, but this is a DNA strand. If you have weak ones, it makes you weak's not the right word. But yes, you're on the right track. So these 3 genes collectively will eventually make an enzyme called lactase, right? Lactase. Remember ACEs and OCEs? If you weren't with me last semester, ACE molecules like lactase are enzymes that break down usually a sugar or some other thing or build something up that has the same name. So lactase is the enzyme that's being produced. You're not making lactose sugar, you're making the enzyme that breaks it down. Are we comfortable with that? Now, there's the operon includes all 5 of these sections. Okay, the promoter, think of that like a landing strip. You know, like when you go to the airport and the airplanes are coming in, the promoter's like, land here, put your flaps down, woo, right here, right? Like right here. That's what it's saying to you. You could look at it like a cheerleader too, like, rah, rah, ree, bind with me. I don't know, whatever works for you. That's the promoter. Nice, jealous. Texas is warm. So the operator says, maybe, right? Like maybe you can go forward and make this protein. Maybe, if I let you. So the operator is the decision maker. Okay, so the operator is going to decide if this whole thing's going to happen. All right, GBPA. I don't know. Leave it be. Move on. Okay. There's a couple of other players to this whole thing, but they're not in this. Are they in this picture? They're not in this picture. So that's the operon. What you need to understand to see how this whole thing works is if this DNA strand were to continue farther on, which it does, of course, but like up here might be a little snippet that codes for something called a repressor, right? So the repressor may hang out on the operator, but it's not in the operon. It's going to be farther up the DNA chain. So the whole DNA is going to make everything it needs for this, okay? Any questions so far? If you want to chime in on the chat and just give me a thumbs up, a yes, a hey, hallelujah, something like that. Okay, cool. If you do, please, I don't always see hands raised, so please be comfortable chiming in the chat if you need me to slow down or repeat something, because sometimes I'll miss hands up. All right, so I'm not going to go over all of this, but I want to go over this part because I used a word in the video, which wasn't wrong. It wasn't an incorrect word, but it wasn't the best word. And I have all the best words, but I didn't this time. It's not an incorrect word. It's just not the one that was used throughout the rest of the lesson. So let's look at this from another perspective. I want you to meet all the players. Now, we all know what an enzyme is, right? An enzyme is a doer. It's a molecule that's going to do work. So this is an enzyme. Do you remember from the video, it's also on the page, what the name of this enzyme is called? Anybody remember what the name of this enzyme is called? In transcription, nope. We're going to make that eventually, Gavin, but first we have to make the mRNA that's going to make that enzyme eventually.
Isn't it like polymerase?
Yes, polymer is a large molecule. Polymer, it's an ACE, means it's an enzyme. And what molecule is made in transcription? Remember, I gave you this example last semester, and I said, imagine you're at the doctor. The doctor has all the information. That's the DNA. But doctors have a complicated verbiage, like they use medical terminology. That's kind of hard to understand. So there's always a person that transcribes what the doctor said and gives it to you to leave the doctor so you can say, oh, I have to take one of these at night, get plenty of rest, have an apple a day, blah, 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 right? In sloppy handwriting, right? So transcription takes the DNA and transcribes it into a message that leaves the nucleus that's more concise, that has the instructions for making just that one protein, not all the proteins you could make. So what is it called? The message that leaves the nucleus in transcription. It's from last semester.
Messenger RNA.
Messenger RNA. Very good. So this molecule, mRNA polymerase, is going to make the message that is what this says, right? Eventually. Maybe, if the operator says it's okay. So that's what this molecule is doing here. Number 2, no, I've had lots of people reading, and that's okay. Somebody else has been chiming in too. So this is mRNA polymerase. This is the promoter. That's where it says, hey, mRNA polymerase, if you want to make a protein, start here. Bind with me. That's the promoter. Then number 2 here is called a repressor. What I said in the video was inhibitor, which is still right, but not awesome. No, I'm totally fine with what's going on. I got no problem with how it's happening. Don't worry about it. So number 2 is AKA an inhibitor, but we're not going to use that term. So that's why I'm taking the time right now. We're going to focus on the word repressor. Okay, then number 4 is what? What is number 4 called? This red section here.
Operator.
The operator. Very good. So the operator is going to decide if it's going to happen. Any visual clues here that's going to decide if it's going to happen? You can compare it, just for comparison's sake, you could compare it to down here. My mom's cat is tearing something up. Just compare the green molecule here to the same green molecule down here. What's different? What's different between scenario A and scenario B?
The repressor is binded to it binded to
Yep.
something and it opened. It turned
Okay.
off.
So in the scenario A, the repressor is bound to the operator, which the operator never moves. It's stuck on the sequence, but the repressor can fall off or come back on. So in scenario B, the repressor is not bonded. Repressor. Anyway, is not bonded to the operator. Do you all see that? That's the important part that I need you to see. To say, yes, I see that, or I don't know what you're talking about. Tell me in chat what we're doing here, or on the mic. Okay, I know that sounds elementary, but that's the crux of this whole complex. If you look at the RNA polymerase in scenario A versus scenario B, the polymerase is bonded to the promoter, and it's going to be moving forward because there's nothing stopping it. But look, it's hitting like a roadblock in scenario A. The RNA polymerase is like physically, chemically cannot pass. You shall not pass, because the repressor is stuck to the operator like a roadblock, like a speed bump, right? So as long as the repressor is sitting here on the operator, as you see it here in scenario A, the RNA polymerase cannot move forward. That's why the operator is the one that decides if it's going to happen or not. If the operator is free, sure, go ahead, pass. You can pass. If it's not, nope. So I guess the operator and repressor together, but really that's what you look at, if there's something stuck to the operator or not. Now, the reason that the repressor is not sitting on the operator in scenario B here is because of this molecule right here. What molecule is this? If you look at our instructions over here, what does it say that is? What molecule is distracting the repressor in scenario B that you don't see in scenario A? Good. Yes, 3 votes for lactose so far. This molecule is lactose. Ose, not ace. What is lactose? Milk and stuff. Let's call it the stuff in milk, right? Good. Thank you, Lucas. It's the sugar in milk. It's not a protein. It's a sugar. It's a carbohydrate. And although it's drawn like this, technically, lactose is a disaccharide. Remember, it looked like this, where you have glucose and galactose, which doesn't matter. You don't have to know those names. There is sugar in milk. So we, as our bodies prefer glucose. This is the paycheck. Remember that we break down and to do all the work in the cell? We can make galactose into glucose later, but glucose is the one we like. We'll run galactose through another system, but there you go. So don't misunderstand big picture. Just step back for a second. There are also proteins in milk. Sometimes babies have an intolerance for proteins in milk, but that doesn't necessarily mean they're lactose intolerant because lactose is a sugar. So, yes, there's everything in milk. Milk has fat, if you get the good stuff. Milk, of course it has milk, it is milk. Fat, sugar, and proteins. Anyway, back to the topic. So, the only way that the enzyme lactase is made is if lactose is around. Let's think about that for a minute. Remember, our bodies strive to be efficient, keep that internal homeostasis balance. You don't want to be making a bunch of proteins and enzymes that have nothing to do. Okay? So if there's no lactose in your system, even if whether you're lactose intolerant or not, if you're not lactose intolerant, if there's no lactose, you won't make lactase because you don't need to. So here's an example. It's all about me. It's not, but for a little bit of time, I was dumb, and I was dumb. I shouldn't say I was dumb. Okay, I had an eating disorder in high school because I had an issue with myself. Oh, 5 minutes. Good. Thanks, Alicia. Long story short, I stopped eating milk, and I developed a temporary lactose intolerance because it took me time to start making those enzymes again, right? So there you go. Now, this is what you have to know. This is what you have to walk away with. If there's lactose present, it'll bind to the repressor, and then lactase will be made. Lactase will be made. But if the enzyme is if polymerase is prohibited, then there will be no lactase because there's no protein production. Good? So I wanted to take a minute and make sure we go through this. Go back and watch the video if you need more. There was a couple of recap questions. That I wanted to go over with you in the few minutes we have left. So I need you to be able to look at an image like this. Okay. And for the test, you've got to be able to answer questions like this. When transcription cannot take place, what is happening? A or B? Is the gene on or is the gene off? Just tell me in chat for the sake of our time. Yeah. Remember, transcription is the first part to get from DNA to mRNA. And if that cannot happen, then the DNA gene is off. Okay, moving on to the next question. What happens when all the lactose is broken down? RNA polymerase binds to the operator. The repressor binds to the operator to block transcription. Transcription continues. We're not going to use D right now. What do you think? A, B, or C? Now, if you said A, no, but that's okay because you're learning. And here's why. RNA polymerase is going to make eventually the lactase. It's responsible for making that happen. So if the RNA polymerase binds to the operator, then you're going to make lactase. But if all the lactose is broken down, you don't need lactase in the system, right? So there's no need for more lactase if it's already broken down. So it's not A. By default, what else is it not? So I guess that's maybe not the most efficient way of asking that question, but what is also not true? Right, because this happens during transcription. I have left no room for myself during transcription. Right, so this one is also false. The answer is B. You're going to block, think of it as off, the gene. Okay? You'll have a matching diagram, something like this, where the operon genes would be what 7 is pointing to. So that's 7. The operon itself would be 6, although it's not awesomely labeled. I might redo this. I did not make this, by the way. RNA polymerase is number 4. That's this molecule here that's bonded to the promoter. mRNA is what's coming off, so that's 5. The repressor is down here, so that's 2. Number 1 is a regulatory gene that will make the repressor later. So that's 1. And then the inducer is 3. It's a fancy way of saying, in this case, lactose. It's not always lactose, but in this case it is. Okay, so I just gave you the answers fast because I wanted you to have them. We are out of time. Make sure that you can do A or B on these, will be on the test. If you need to double check them, the answers will be in the master guided notes. And again, when you watch, if you need to learn more about lactose intolerance, you can answer them. There's answers here will also be in the guided notes. So this was good.